Our world is full of life. From the tiniest of bacteria to the largest whale, we share our planet with millions of species. But the Earth is always changing, and species come and go naturally over long periods of time. However, in the case of the 5 mass extinctions, the Earth lost more than 75% of its species in a very short amount of time. And scientists now have evidence that we, human beings, are on the brink of causing another one, the 6th mass extinction in our planet's history. So what's being done? Conservation biologists have long worked to gather data by monitoring the health of both populations and individuals. But a relatively new player has entered the game of conservation biology, and its name is deoxyribonucleic acid, or DNA. At the University of Montana, scientists like Dr. Gordon Leuckart are working hard to incorporate molecular data into analyzing at-risk wildlife populations. My next stop was the biological field station at Flathead Lake, where I could talk to the geneticists who worked in the field.
Conservation genetics is the use of genetic tools to solve problems in conservation. There are a wide range of questions that genetics can answer. One of the simplest is, how much does genetic variation matter to an individual's performance?
I figured the best way to understand this field was to learn about the kinds of studies being done.
And recently we have a lot of research on cutthroat trout, which are native, and rainbow trout, which are introduced invasive pests. Hybridization is problematic because if you bring in genes from another species and they hybridize, usually the fitness of the individuals is reduced compared to the pure native individuals. And that's a problem because then we'll have fewer fish. So it's important to identify hybrid fish and the spread of hybridization through river networks.
I couldn't help but ask why we really needed to be doing this kind of work. What information can we get using molecular data that we couldn't get elsewhere?
It's very difficult to tell if an individual has a small proportion of genes from another species. So if the Westlope cutthroat trout has genes from the rainbow trout, you often could not tell and you would get the answer wrong. With simple genetic tests, you can identify if an individual has 1% of its genome from the non-native rainbow trout.
In order to study the effects of hybridization, hybrid fish have to be identified. That's where the genetics comes in. Take these 2 fish. Their differences on the outside are no more than you'd expect between any 2 individuals from the same species. However, the DNA tells a different story. Deep inside of their genomes, there are tiny differences in their sequences. Only genetics can reveal that the fish on the bottom is actually a hybrid with another species.
Montana has often led the nation in using genetics for trout management. They want to know the answer because they'll use the information to adapt their management strategies based on what the science says.
It seemed like conservation genetics is the perfect blend of all the things a scientist could ask for in a project. But how important is the work that's being done in the grand scheme of things?
What's the meaning of life? Well, it's in DNA. Without genetic variation, there will not be adaptation. But one of the most urgent issues for society today is conserving biodiversity, understanding what we're losing before it's gone. We are expected to lose 20 or 30% of all named species on the planet in the next few decades. So in a way, you could argue there's no more important discipline today than conservation genetics. As a student, wanting to do fun and exciting work and handle animals, but also make a meaningful contribution to society, help save the world, help save humanity from this massive extinction crisis, but a real rewarding and promising area is applying genomics. It's one of the most exciting areas in all of science.
So what kind of science interests you? Maybe conservation genetics seems like the perfect fit, or maybe not, but that's not the point. The point is that science is changing every day, and the questions we'll be able to answer tomorrow may never even seem possible today. You may not ever know where science will go next, but at least you'll be along for the ride.